Hello viewers, welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share and subscribe. Let's start!
In the previous video, we have learned about restriction enzymes. And today we are going to continue with types of restriction enzymes. So what did we learn in the previous video? We learned about what are restriction enzymes, what are recognition sites, what are blunt ends, staggered ends, frequency of restriction enzymes, and on the basis of how it is cutting at the recognition site, what are the different types of restriction enzymes. Now we are going to learn properly about the restriction enzyme types. So let's just revise the basics one. So what is a restriction enzyme? It is an endonuclease that recognize short sequences which are palindromic. Palindromic means they are the mirror images, okay, of 4 to 8 base pairs, and in the presence of magnesium, they cleave the DNA. In the close proximity, that means if this is the restriction site, they may be cleaving it here. If this is the restriction site, they may be cleaving it here. And if it's the restriction site, they may be cleaving it somewhere beyond a distant place, okay, at a random place. So this is how the restriction enzyme works. Now, as we learned in the previous video even, that this restriction enzyme was first observed in bacteria, isn't it? So what are they doing? How does it work there? So what happens is these bacteria or archaea, they protect themselves from the lethal phage infections, bacteriophage infections. So what happens? They use a mechanism called restriction modification system. For example, this is a bacteriophage. Okay, now this bacteriophage has its own DNA, but when it enters the host cell, this is the DNA of the host cell. Now, for the host cell, the bacteriophage DNA is a foreign DNA, isn't it? Now, what does the host cell do is, it recognizes a particular site in the foreign DNA, for example, GATC, and it cleaves it there. That means, now what is this doing? It is cleaving or breaking the foreign DNA, and if the foreign DNA is broken down, it won't infect the bacterial cell. Now, this is the first thing. See, here it is written dual activity. So we just learned about the first thing, that is, it is inactivating by generating the double-stranded base. Now the question arises, what if the same recognition site is present in the host cell? Okay, host DNA. Why is it not cleaving its own DNA? It's not cleaving its own DNA because it is methylating it. Okay, and when I say it's methylating the DNA, that means it does not allow the restriction enzymes to act upon it. Okay, it's just like a blanket. Okay, so it does not allow the restrictions, enzymes cannot recognize it, and thus they don't cleave it. So this is what is restriction enzyme, and this is why we are using it in the recombinant DNA technology. Okay, because the main important process of recombinant DNA technology is cleaving the DNA at a particular site. Okay, now we are going to learn the types of restriction enzymes. See, generally there are 4 types. Okay, but if you talk about in a detailed manner, it is a 3 type. Okay, the 4 type is just a new one. In some places you don't find type 4. In some places you find type 4. Okay, just for your reference, I have given here 4 types. Now, how are we characterizing it? The first thing is we characterize it on the basis of its composition. And then what are its cofactors? For example, for some it needs ADP, it hydrolyzes ATP. For some it does not hydrolyze ATP. Okay, so that's how the cofactor changes. Now, what is the target sequence and the position where it is cleaving? So just now I just showed you that if this is the recognition site or restriction site, it may cleave here. If this is a recognition site, it may cleave at some distance. If this is it may cleave at a very distance, that is far away from the recognition site. So based on that also, we are going to differentiate it. Okay, this is one of the most important characterization. Okay, so let's learn one by one. The first one we are going to learn is type 1 restriction enzyme. In today's video, we are only going to deal about type 1 restriction enzyme. In further videos, we'll be learning about type 2, type 3, and type 4. So what is type 1 restriction enzyme? Type 1 restriction enzyme is first identified in the strains of E. coli. Okay, and these enzymes, where does it cut? It cut at a random distance. It may be at least 1,000 base pairs away from the restriction site. Okay. Now, this type 1 restriction enzyme is encoded by 3 genes. We call it as HSD, that is host specificity determinant. Now, if it is having R, then it means it encodes for restriction. If it is having a subunit of M, it encodes for modification. And if it has a subunit of S, it encodes for specificity. Okay, that's how it works. Okay, now, see, now this restriction enzyme, type 1 restriction enzyme, it forms a pentameric complex. So what does a pentameric complex means? The pentameric complex means it consists of two restriction endonuclease, two methylase transferase proteins, and one specificity protein. Okay, so this is how it forms a complex. Now, modification. Where does this modification occur? So this modification, it occurs at the recognition site, and they require as adenosyl methionine. It requires adenosine methionine. Okay, but cleavage, just now we learned it. Where does the cleavage occurs? The cleavage occurs at a random site, nearly 1,000 base pairs. Okay, and it requires adenomethionine and ATP. It requires adenomethionine and ATP. It requires both of it for the cleavage. Okay, now let's learn how it is cleaving. For example, this is a double-stranded DNA, and this is where our recognition site is and the restriction enzyme binded here. Now, what happens? See, once this protein complex binds here, it forms a molecular motor. Okay, and it uses ATP to translocate the bases. Okay, so here this is how the bases start forming. Okay, it's translocating the bases. This is how it forms. So till where it translocates the bases? It translocates the bases until and unless it reaches the second complex. So you have two complexes, so it reaches the second complex, it translocates the bases. Now, once it reaches the second complex, it's gonna cleave the double-stranded bonds. Okay, this is how the type 1 restriction enzyme works. Okay, I think it's very clear. Now, type 1 restriction enzymes are very important biochemical agents. Okay, we use it there. But practically, it has very less value because they do not produce distinct gel banding fragments. We need distinct fragments, so it does not produce distinct gel banding fragments. So we don't use a restriction enzyme type 1 a lot. Okay, so this was all about restriction enzyme type 1. In the next video, we are going to learn restriction enzyme type 2 and its subtypes. So restriction enzyme type 2 has to be more detailed because it has a lot of subtypes. And then in the another video, we are going to learn restriction enzyme type 3 and type 4.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to Biodrunk. Let's meet in the next video. Bye-bye.